Hello everyone. So with the help of this video we are going to learn about DNA replication, some basic ideas about this DNA replication process, semi-discontinuous synthesis like Okazaki fragments, replication origin and replication fork, unit of replication, extra chromosomal replication of bacterial TA plasmid along with enzymes or proteins of replication like primases, replisomes, helicase, DNA polymerases, single-strand binding proteins, topoisomerases and ligases, then fidelity of replication, then replication of the ends of eukaryotic chromosome, the role of telomerase, then models of DNA replication, rolling circle model and looped rolling circle model, D-loop model, theta model, then inhibitors of DNA replication like methotrexate and fluorodeoxyuridylate. So let's start learning. So when we learn molecular biology, we often think what is the importance of this topic? What is the importance of learning molecular biology? What are the advantages and disadvantages and applications of molecular biology? So let's start learning after recognizing the purpose of the efforts to learn. So molecular biology is the branch of biology that deals with the study of biological activity at the molecular level. It involves the study of the structure and function of macromolecules such as DNA, RNA and proteins and how they interact within and between cells. So here are some of the importance, advantages, disadvantages and applications of molecular biology, like understanding of life processes. That is a major important point. Understanding of life processes. Molecular biology has allowed scientists to understand the integrate processes that govern life, including gene expression, DNA replication and protein synthesis. Then advancement in medicine. Molecular biology has contributed to the development of various medical applications, including diagnostic tests, vaccines, and gene therapies. Then, development of biotechnology. Molecular biology has been instrumental in the development of biotechnology. Molecular biology has been instrumental in the development of biotechnology, which has allowed for the production of novel drugs, food products, and industrial chemicals. Then precision and accuracy. Molecular biology techniques are highly specific and precise, allowing for the detection of even small amounts of genetic material. Then speed. Many molecular biology techniques are rapid, allowing for the analysis of large numbers of samples in a short period. Then versatility. Molecular biology techniques can be applied to a wide range of biological systems and samples, from bacteria to human tissues. There are some disadvantages also, like cost. Many molecular biology techniques require expensive equipment such as reagents, which can be a barrier to their use. Then complexity. Molecular biology techniques often involve complex protocols and require skilled technicians to perform. Then ethical concerns. Molecular biology techniques, particularly those involving genetic engineering, raise ethical concerns regarding their potential impact on society and the environment. Then applications. Disease diagnosis. Molecular biology techniques are used for the detection of pathogens and genetic mutations associated with diseases. Then forensics. Molecular biology techniques are used in forensic investigations to identify suspects and victims and to establish the presence of biological material at a crime scene. Then genetic engineering. Molecular biology techniques are used to modify the genetic material of organisms for research or commercial purposes, including the production of genetically modified crops and animals. So, in conclusion, molecular biology has had a profound impact on our understanding of life processes, and its applications in medicine, biotechnology, and forensics have revolutionized these fields. However, the use of molecular biology techniques also raises ethical concerns, and their cost and complexity can be a barrier to their widespread use. Then, first topic of DNA replication, like semi-discontinuous synthesis. Okazaki fragments. Semi-discontinuous DNA synthesis is a mechanism by which the lagging strand of DNA is synthesized during DNA replication. The synthesis of the lagging strand is discontinuous because it is synthesized in short fragments like Okazaki fragments. So the lagging strand of DNA is synthesized in the opposite direction of the replication fork movement. Therefore, the synthesis of the lagging strand occurs in small fragments of DNA ranging from 1000 to 2000 nucleotides in length. So we just learned the lagging strand of DNA is synthesized in the opposite direction of the replication fork movement. So replication fork movement is in one direction, but the synthesized DNA is produced, that is a lagging strand is produced in the opposite direction. So therefore the synthesis of the lagging strand occurs in small fragments of DNA ranging from 1000 to 2000 nucleotides in length. These fragments are called Okazaki fragments and are synthesized in the 5' to 3' direction. Mechanism. The semi-discontinuous DNA synthesis mechanism involves 3 steps like initiation, elongation and termination. First step, initiation. The initiation of DNA synthesis on the lagging strand occurs after the primase enzyme synthesizes a short RNA primer. The RNA primer provides a free 3' OH group for DNA polymerase 3 to add nucleotides in the 5' to 3' direction. The leading strand is synthesized continuously in the same direction as the replication fork movement. Then elongation. After the DNA primer is synthesized, DNA polymerase 3 binds to the primer template junction and begins to synthesize the first Okazaki fragment. As DNA polymerase 3 adds nucleotides to the 3' OH end of the RNA primer, it displaces the RNA primer, creating a gap in the newly synthesized DNA strand. So the RNA primer is then removed by the RNase enzyme and the resulting gap is filled in by DNA polymerase 1 using the 3' OH end of the adjacent Okazaki fragment as a primer. So this process of gap filling and Okazaki fragment synthesis continues until the lagging strand is fully synthesized. Then termination. Once the lagging strand is fully synthesized, the gaps between Okazaki fragments are sealed by DNA ligase, which catalyzes the formation of a phosphodiester bond between adjacent nucleotides. Then major important points about this topic, like semi-discontinuous DNA synthesis occurs on the lagging strand of DNA during replication. The lagging strand is synthesized in short fragments called Okazaki fragments. The synthesis of the leading strand is continuous in the 5' to 3' direction. The initiation of DNA synthesis on the lagging strand requires the synthesis of an RNA primer by primase. The synthesis of the Okazaki fragments requires the coordinated action of DNA polymerase 3, RNase H, and DNA polymerase 1. The gaps between Okazaki fragments are sealed by DNA ligase. So in conclusion, semi-discontinuous DNA synthesis is a crucial mechanism in DNA replication that allows for the efficient and accurate synthesis of both the leading and lagging strands of DNA. The synthesis of Okazaki fragments is essential to ensure the continuity of the lagging strand and is a result of the opposite direction of DNA synthesis on the lagging strand compared to the leading strand. Then next topic, replication origin and replication fork. So replication origin and replication fork are two critical components of the DNA replication process. So the replication origin is a specific sequence of nucleotides in DNA where DNA replication begins. It is recognized by the replication initiation proteins that bind to the origin and recruit the DNA helicase enzyme to initiate DNA replication. So in bacteria, the replication origin is a single site on the circular chromosome. In eukaryotes, the replication origin is much more complex and consistent of multiple sites that are recognized by different proteins. The replication origin is essential for the initiation of DNA replication and the coordination of the replication machinery. Then in bacteria, the replication origin is called ORIC and it is a 245 base pair sequence located at a specific site on the circular chromosome. In humans, the replication origin is a complex sequence that consists of multiple sites located throughout the genome. The replication origin in humans is essential for the initiation of DNA replication and the maintenance of genome stability. Later importance, the replication origin is essential for the initiation of DNA replication and the coordination of the replication machinery. The recognition of the origin by replication initiation proteins ensures that replication occurs on a controlled and regulated manner. The replication origin also plays a critical role in maintaining genome stability by ensuring that each chromosome is replicated once and only once during each cell division cycle. Then replication fork. The replication fork is the site where DNA replication occurs. It is formed by the unwinding of the double helix by a DNA helicase enzyme, which separates the two strands of DNA. The replication fork is composed of two strands, the leading strand and the lagging strand. The leading strand is synthesized continuously in the 5' to 3' direction, whereas the lagging strand is synthesized in short fragments called Okazaki fragments in a semi-discontinuous manner. The replication fork is essential for the efficient and accurate synthesis of new DNA strands during DNA replication. So, for example, in bacteria, the replication fork is a single site on the circular chromosome where DNA replication occurs. In eukaryotes, the replication fork is much more complex and consists of multiple sites where DNA replication occurs simultaneously. The replication fork in eukaryotes is essential for the efficient and accurate synthesis of new DNA strands during DNA replication. So, what is the importance? The replication fork is essential for the efficient and accurate synthesis of new DNA strands during DNA replication. The leading and lagging strands are synthesized simultaneously at the replication fork, allowing for the efficient duplication of the genome. The replication fork also plays a critical role in maintaining genome stability by preventing the accumulation of DNA damage and ensuring the faithful transmission of genetic information to daughter cells. So, in conclusion, replication origin and replication fork are two critical components of the DNA replication process. The replication origin is essential for the initiation of DNA replication and the coordination of the replication machinery, while the replication fork is essential for the efficient and accurate synthesis of new DNA strands during DNA replication. So, understanding the structure and function of the replication origin and replication fork is essential for understanding the process of DNA replication and the maintenance of genome stability. Then, unit of replication, extra chromosomal replicant of bacterial TA plasmid. Unit of replication and extra chromosomal replicant are two important concepts related to bacterial genetics. So, unit of replication. The unit of replication is the segment of DNA. Then, unit of replication. The unit of replication is the segment of DNA that is replicated from a single origin of replication. In bacteria, the unit of replication is often referred to as a replicon, and it includes the DNA sequence that is replicated from a single origin of replication to the point where the replication machinery meets at the opposite end of the replicant. The size of the replicant can vary depending on the organism, and it can range from a few thousand base pairs to millions of base pairs. For example, in bacteria, the unit of replication can be a plasmid or a segment of the chromosome. In the case of Escherichia coli bacterium, the chromosome has multiple origins of replication, and each of these origins represents a unit of replication. So, additionally, bacterial plasmids such as the F plasmid also have a single origin of replication that serves as the unit of replication. The concept of the unit of replication is important in the study of bacterial genetics as it helps us understand how bacterial DNA is replicated. By identifying the origin of replication and the reduction of replication, we can study the replication process in more detail and gain insights into the mechanisms that govern DNA replication. Then, extra chromosomal replicant of bacterial TA plasmid. The bacterial TA plasmid is a large circular DNA molecule that is found in Agrobacterium tumefaciens. This plasmid has an extra chromosomal replicant that allows it to replicate independently of the bacterial chromosome. The extra chromosomal replicant of the TA plasmid is known as the virulence region, and it contains all the genes required for the transfer of the plasmid into the plant host. So, procedure: the extra chromosomal replicant of the TA plasmid is replicated in a similar manner to the chromosomal DNA. The replication process is initiated at a specific site on the plasmid known as the origin of replication, and the plasmid is replicated bidirectionally from the site. The replicant is then partitioned into the daughter cells during cell division, allowing for the stable maintenance of the plasmid within the bacterial population. So, once again, let's learn about this. The bacterial TA plasmid is a large circular DNA molecule that is found in Agrobacterium tumefaciens. This plasmid has an extra chromosomal replicant that allows it to replicate independently of the bacterial chromosome. The extra chromosomal replicant of the TA plasmid is known as the virulence region, and it contains all the genes required for the transfer of the plasmid into the plant host. So procedure, the extra chromosomal replicant of the TA plasmid is replicated in a similar manner to the chromosomal DNA. The replication process is initiated at a specific site on the plasmid known as the origin of replication, and the plasmid is replicated bidirectionally from the site. The replicant is then partitioned into the daughter cells during cell division, allowing for the stable maintenance of the plasmid within the bacterial population. The TA plasmid is commonly used as a vector in genetic engineering as it can transfer genes into plant cells. So by inserting genes of interest into the VIR region of the plasmid or virulence region, researchers can create genetically modified plants and that express new traits, VIR region, and create genetically modified plants that express new traits such as resistance to herbicides or increased yield. The extra chromosomal replicant of the TA plasmid is important in genetic engineering as it allows for the stable maintenance of the plasmid within bacterial populations. This makes it an attractive tool for the transfer of genes into plant cells, allowing for the creation of genetically modified plants with new traits. Additionally, the study of the TA plasmid has helped us understand the mechanisms that govern bacterial plasmid replication and the transfer of DNA between bacterial cells. So in conclusion, the unit of replication and extra chromosomal replicant are two important concepts in bacterial genetics. The unit of replication is a segment of DNA that is replicated from a single origin of replication, while the extra chromosomal replicant is the DNA segment that allows a plasmid to replicate independently at the bacterial chromosome. So understanding these concepts is important in the study of bacterial genetics and development of genetic engineering tools. Then enzymes, proteins of replication. Enzymes and proteins are key players in the process of DNA replication. DNA helicase is a protein that unwinds the double helix structure of the DNA molecule. The enzyme breaks the hydrogen bonds and hold the two strands together, allowing the DNA strands to separate and form a replication fork. For example, E. coli DNA ligase is known as the DNAB, while the human DNA helicase is known as MCM, mini chromosome maintenance protein. DNA helicase plays a crucial role in the initiation of DNA replication. Without this enzyme, the DNA strands would remain stuck together and replication could not occur. Then DNA polymerase. DNA polymerase is an enzyme that adds nucleotides to the growing DNA chain. Earlier we learned about DNA helicase. DNA helicase is a protein that unwinds the double helix structure of DNA molecule. The enzyme breaks the hydrogen bonds that hold the two strands together, allowing the DNA strands to separate and form a replication fork. E. coli DNA helicase is known as DNAB, while the human DNA helicase is known as MCM, like mini chromosome maintenance protein. DNA helicase plays a crucial role in the initiation of DNA replication. Without this enzyme, the DNA strands would remain stuck together and replication could not occur. Then DNA polymerase. DNA polymerase is an enzyme that adds nucleotides to the growing DNA chain. The enzymes that can only add nucleotides in the 5' to 3' direction, meaning that it can only add nucleotides to the free 3' end of the DNA chain. For example, E. coli DNA polymerase is known as DNA pol 3, while the human DNA polymerase is known as pol alpha beta, like that. DNA polymerase is essential for the replication of DNA. It adds nucleotides to the growing chain, ensuring that the new DNA strand is a fruitful copy of the original DNA strand. Then DNA ligase. DNA ligase is an enzyme that catalyzes the formation of a phosphodiester bond between adjacent nucleotides in the DNA strand. The enzyme is responsible for sealing the gaps in the lagging strand of the DNA molecule where the Okazaki fragments have been synthesized. For example, E. coli DNA ligase is known as Lig-A, while the human DNA ligase is known as Lig-1, Lig-3 and Lig-4. DNA ligase is important for the completion of DNA replication. Without this enzyme, the Okazaki fragments in the lagging strand would remain unconnected and DNA strand would be incomplete. Then single-stranded DNA binding proteins. Single-stranded DNA binding proteins are proteins that bind to the single-stranded DNA that is formed during the replication process. The proteins prevent the DNA strands from reannealing, ensuring that the DNA remains in its single-stranded state. Example, E. coli single-stranded DNA binding protein is known as SSB, while the human single-stranded DNA binding protein is known as RPA, replication protein A. Replication protein A. Important, single-stranded DNA binding proteins play a crucial role in the replication process. They prevent the DNA strands from reannealing. That is replication protein A. Single-stranded DNA binding proteins play a crucial role in the replication process. They prevent the DNA strands from reannealing, which would disrupt the replication process. Then primase is there. Primase is an enzyme that synthesizes short RNA primers on the lagging strand of the DNA molecule. The RNA primers provide a free 3' end for the DNA polymerase to start synthesizing the Okazaki fragments. Examples, E. coli primase is known as DNAG, while the human primase is known as PRIM1 and PRIM2. Primase is important for the initiation of DNA synthesis on the lagging strand. Without the RNA primers, the DNA polymerase would have no free 3' end to start synthesizing the Okazaki fragments. So in conclusion, enzymes and proteins play crucial roles in processing of DNA replication. DNA helicase unwinds the double helix. DNA polymerase adds nucleotides to the growing DNA chain. DNA ligase seals the gaps in the lagging strands. Then primase in detail. Primase is an enzyme that plays a crucial role in the replication of DNA. Definition of primase. Primase is an RNA polymerase enzyme that synthesizes short RNA primers on the lagging strand of DNA molecule. The RNA primers provide a free 3' end for the DNA polymerase to start synthesizing the Okazaki fragments. Then procedure of primase. The primase enzyme works together with DNA helicase and DNA polymerase to initiate DNA replication. The primase synthesizes a short RNA primer that binds to the template strand for the DNA molecule. The RNA primer provides a free 3' OH group that the DNA polymerase can attach to, initiating the synthesis of Okazaki fragments on the lagging strand. Once the RNA primer has been synthesized, it is later removed and replaced with DNA nucleotides by the DNA polymerase. Then some examples of primase. Primase is found in both prokaryotes and eukaryotes, and its amino acid sequence is highly conserved across different organisms. Examples of primase include DNAG in E. coli and PRIM1 and PRIM2 in humans. Then importance of primase. Primase is a crucial enzyme in the DNA replication because it is responsible for the initiation of DNA synthesis on the lagging strand. Without the RNA primers synthesized by primase, the DNA polymerase would not have a free 3' OH group to start synthesizing the Okazaki fragments. As a result, DNA replication would not occur, leading to errors in the DNA sequences and potentially causing cell death or disease. Then regulation of primase. Primase activity is tightly regulated to ensure that DNA replication proceeds smoothly and without errors. The activity of primase is coordinated with other enzymes and proteins involved in replication, such as the DNA helicase and DNA polymerase. The timing and amount of primase activity are also regulated by the cell cycle, as the DNA replication only occurs during specific phases of the cell cycle. So in conclusion, primase is an essential enzyme in DNA replication, synthesizing short RNA primers on the lagging strand that provide a free 3' OH group for the DNA polymerase to start synthesizing the Okazaki fragments. The regulation of primase activity is important for the accurate and efficient replication of DNA, ensuring the integrity of genetic material and preventing the development of diseases such as cancer. Then replisomism. Replisomes are multi-protein complexes that play a central role in the process of DNA replication. Replisomes are large protein complexes that are responsible for DNA replication. They consist of several different proteins, including DNA polymerases, helicases, primases, and other accessory proteins. Then procedure of replisomes. During DNA replication, the replisomes binds to the DNA molecule at the replication origin and begins to unwind the double-stranded DNA using helicase enzymes. Once the DNA strands are separated, the replisome complex coordinates the activity of the different enzymes involved in DNA replication. The leading strand is synthesized continuously by the DNA polymerase, while the lagging strand is synthesized in short fragments by the DNA polymerase with the help of RNA primer synthesized by primase. The DNA fragments are later joined together by the enzyme DNA ligase. Then examples of replisomes. Replisomes are found in both prokaryotes and eukaryotes. In bacteria, the replisome is composed of DNA polymerase 3 holoenzyme, helicase, primase, and other accessory proteins. In eukaryotes, the replisome is composed of several different polymerases, including pol alpha, like that, as helicases, primases, and other accessory proteins. Then importance of replisomes. Replisomes are essential for the accurate and efficient replication of DNA. They coordinate the activity of the different enzymes involved in DNA replication, ensuring that DNA is copied with high fidelity. So, in addition, replisomes play a crucial role in the maintenance of genome, as errors during DNA replication can lead to mutations and potentially cause diseases such as cancer. Then, regulation of replisomes. The activity of the replizome is tightly regulated to ensure the accurate and efficient replication of DNA. The timing and amount of replisome activity are controlled by the cell cycle, as DNA replication only occurs during specific phases of cell cycle. In addition, the activity of the replisome is coordinated with other proteins and enzymes involved in DNA replication to ensure that the DNA is copied correctly. So, in conclusion, replisomes are large protein complexes that are responsible for the accurate and efficient replication of DNA. They coordinate the activity of different enzymes involved in DNA replication and play a crucial role in maintaining the integrity of the genome. The regulation of replisome activity is important for ensuring the accuracy of DNA replication and preventing the development of diseases such as cancer. Then helicase in detail. Helicase is an enzyme that plays a crucial role in DNA replication and repair. Helicase is an enzyme that uses energy from ATP hydrolysis to unwind and separate the double-stranded DNA molecule into single strands. This allows the DNA polymerase to access the single-stranded template for replication or repair. Then procedure of helicase. During DNA replication, helicase binds to the DNA molecule at the replication origin and begins to unwind the double-stranded DNA. It does this by breaking the hydrogen bonds that hold the two strands together, and it moves along the DNA molecule in a process called translocation. As the helicase moves along the DNA molecule, it continues to unwind the double-stranded DNA, creating a replication fork. The replication fork is the point at which the double-stranded DNA is being separated into single strands and is the site of active DNA replication. Then some examples of helicase. Helicase is found in both prokaryotes and eukaryotes. In bacteria, the helicase is composed of a single protein, whereas in eukaryotes it is composed of several different proteins. Then examples of helicase include the E. coli helicase DNAB and the human helicase BLM. Importance of helicase. Helicase plays a crucial role in DNA replication and repair. Without helicase, the DNA strands would remain tightly wound together and DNA polymerase would not be able to access the template strand for replication. Then helicase also plays a role in DNA repair, as it can unwind the DNA molecule to allow repair enzymes to access damaged DNA strands. Then regulation of helicase. The activity of helicase is regulated by a variety of factors, including other proteins and enzymes involved in DNA replication and repair. Helicase activity is also coordinated with the activity of other proteins such as DNA polymerase to ensure that the DNA is replicated accurately and efficiently. So, in conclusion, helicase is an enzyme that plays a crucial role in DNA replication and repair. It unwinds the double-stranded DNA molecule, creating a replication fork and allowing DNA polymerase to access the template strand for replication. Helicase is found in both prokaryotes and eukaryotes and is regulated by other proteins and enzymes involved in DNA replication and repair. Without helicase, accurate and efficient DNA replication would not be possible. Then DNA polymerases. DNA polymerases are enzymes that play a critical role in DNA replication, repair, and recombination. Here is a detailed explanation of different types of DNA polymerases, their major points, examples, and importance. First, DNA polymerase 1. DNA polymerase 1, Pol 1 is a prokaryotic DNA polymerase that was the first to be discovered. Pol 1 has a 5' 3' polymerase activity as well as a 3' 5' exonuclease activity that is involved in proofreading and repair. Pol 1 is essential for DNA replication and repair in bacteria, and it also plays a role in genetic recombination. Then examples of bacteria that use Pol 1 include E. coli and Bacillus subtilis. Then DNA polymerase 2. DNA polymerase 2. DNA polymerase 2 is another prokaryotic DNA polymerase that is involved in DNA repair. Pol 2 has a 5' 3' polymerase activity and a 3' 5' exonuclease activity. Similar to Pol 1, Pol 2 is induced by DNA damage and is involved in error-prone repair mechanisms. Examples of bacteria that use Pol 2 include E. coli and Bacillus subtilis. Then DNA polymerase 3. DNA polymerase 3 is the primary DNA polymerase involved in DNA replication in prokaryotes. Pol 3 is a complex enzyme composed of several subunits including the catalytic core, the clamp loader complex, and the sliding clamp. Pol 3 has a 5' 3' polymerase activity and a 3' 5' exonuclease activity for proofreading and repair. Examples of bacteria that use Pol 3 include E. coli and Bacillus subtilis. Then DNA polymerase 4. DNA polymerase 4 is a prokaryotic DNA polymerase that is involved in DNA repair. Pol 4 has a 5' 3' polymerase activity and is involved in error-prone repair mechanisms. Pol 4 is induced by DNA damage and is less accurate than other DNA polymerases. Then examples of bacteria that use Pol 4 include E. coli and Bacillus subtilis. Later DNA polymerase 5. Then DNA polymerase 5. DNA polymerase 5 is a prokaryotic DNA polymerase that is also involved in DNA repair. Pol 4 is a 5' 3' polymerase activity and is involved in error-prone repair mechanisms. Pol 5 is induced by DNA damage and is less accurate than other DNA polymerases. Examples of bacteria that use Pol 5 include E. coli and Bacillus subtilis. Then DNA polymerase alpha. DNA polymerase alpha is a eukaryotic DNA polymerase that is involved in DNA replication. Polymerase alpha has a 5' 3' polymerase activity and a primase activity which synthesizes RNA primers that are required for DNA synthesis. Pol alpha also plays a role in DNA repair. Examples of organisms that use Pol alpha include humans and yeast. Then DNA polymerase delta. DNA polymerase delta is a eukaryotic DNA polymerase that is involved in DNA replication and repair. Pol delta has a 5' 3' polymerase activity and a 3' 5' exonuclease activity for proofreading and repair. Pol delta also participate in DNA repair processes including base excision repair and nucleotide excision repair. Examples of organisms that use Pol delta include humans and yeast. Then DNA polymerase epsilon. DNA polymerase epsilon is a eukaryotic DNA polymerase that is involved in DNA replication and repair. Pol epsilon has a 5' 3' polymerase activity and a 3' 5' exonuclease activity for proofreading. So DNA polymerase are a group of enzymes that play a critical role in DNA replication, repair and recombination. These enzymes catalyze the synthesis of DNA from deoxynucleoside triphosphatase like DNPPs using DNA template strand. There are prokaryotic DNA polymerases. Prokaryotic DNA polymerases include DNA polymerase 1, 2, 3, 4 and 5. They are all composed of several subunits and have a 5' 3' polymerase activity and a 3' 5' exonuclease activity for proofreading and repair. DNA polymerase 3 is the primary enzyme involved in DNA replication in prokaryotes, while Pol 3, 1, 2, 4 and 5 are involved in DNA repair. These enzymes are essential for maintaining genome stability and genetic diversity in prokaryotic organisms. Then eukaryotic DNA polymerases are there. Eukaryotic DNA polymerases include DNA polymerase alpha, delta and epsilon. Pol alpha is involved in DNA replication and has a primase activity which synthesizes RNA primers required for DNA synthesis. Pol delta and epsilon are involved in DNA replication and repair and have 5' 3' polymerase activity and 3' 5' exonuclease activity for proofreading and repair. These enzymes are essential for the accurate and efficient replication and maintenance of eukaryotic genomes. Later, structure of DNA polymerases. Most DNA polymerases have a similar structure consisting of a catalytic domain is there, a DNTP binding site, a template binding site, and a proofreading domain. The catalytic domain contains the active site responsible for catalyzing the polymerization reaction, while the proofreading domain contains the exonuclease activity responsible for proofreading and repair. Then procedure for DNA polymerase. During DNA replication, DNA polymerase use a DNA template strand to synthesize a new complementary DNA strand. The polymerase first recognizes the origin of replication and binds to the template strand. The polymerase then adds nucleotides to the 3' end of the primer using DNTPs that are complementary to the template strand. The polymerase moves along the template strand in a 3', 5' direction, synthesizing a new DNA strand in a 5', 3' direction. Then examples of DNA polymerase. Examples of DNA polymerase include Taq polymerase, a DNA polymerase isolated from Thermus aquaticus that is commonly used in polymerase chain reaction, and the PHI-29 polymerase, a DNA polymerase isolated from bacteriophage 529 that is used for whole genome amplification. Then importance of DNA polymerase. DNA polymerase are essential for the replication and maintenance of the genome. These enzymes ensure that accurate and efficient replication of DNA while also maintaining genome stability through proofreading and repair mechanisms. DNA polymerases are also involved in DNA repair and mechanisms, including ensuring genetic diversity and adaptation in different environments. Next topics will be dealt with the help of next video. Thank you for listening.